guys to understand some of the key terms and concepts of genetic engineering, including DNA restriction enzymes and CRISPR, which you probably heard of before. And then understand the applications of CRISPR in gene editing and scientific research, like how it's going to be used and why it's important. And then to consider the ethical concerns around applications of genetic engineering in humans and animals. One of your key points in a row. Gretchen, do you want to start? What's your key point about genes?
Okay, it uses CRISPR as a
Yeah, CRISPR is a technique that can be used to edit genes or answer genes. Yeah.
You said that genes can be turned on.
A type of transformation or editing gene engineering would be bacterial transformation with you inject plasmids.
Yeah.
Yeah, we did that in this class.
We said that, well, genes or DNA is basically the blueprint for the entire body or organism.
And genes give traits to organisms. Science.
See the sign? There's the right on it.
Oh, my bad. Forgive me. Oh, whoops. No, it's over here. No. I'm wrong again. It's over here. Nice. Is there anything else?
And restriction enzymes cut the DNA into...
3 pieces.
Yeah, they cut it into pieces. It doesn't necessarily have to be 3, but yeah, pieces or fragments, any word like that would work. Do you think that restriction enzymes could be used to cut DNA from other organisms?
Yep.
Okay. Why?
Because other organisms have DNA too, so why can't you cut their DNA?
Exactly, same genetic code.
Yep.
Yeah, it has the same structure.
Has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas-9. Like previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Manipulating it, so they choose the guide RNA, and that corresponds to the gene that they want to cut.
Yeah, so they're introducing a segment of RNA that the Cas nuclease will recognize. Okay, so on the whiteboards, brainstorm and record potential pros and cons of genetic engineering. So I know you guys have talked about this already, but it was more focused on plants as far as I know. So if you could talk specifically about humans.
There's also a lot of ethical dilemmas, like there's not really a clear line. So as it was talking about in the video, is it okay to go in and change the DNA of a live organism to see if it has an effect? Like, I don't think anyone has a clear answer on whether or not that's ethical.
I just think it's kind of unethical to like design your babies before they're born and like choose like their traits and stuff. I think that you just shouldn't be doing that. Messing with that.
I just have a question. So CRISPR itself is pretty, like the process is pretty inexpensive in itself. Would it, like, if it got to the point where people could design their own babies, would that be affordable for everyone? Like, there's also a question of the ethics of like how many people can actually afford it.
I think just going off on that, you could potentially change the way life as we know it works. Like if it got to the point where we're designing our own babies, then
Curing genetic disease before babies are born. If there's a focus on making it accessible to everyone.
So now we're gonna have you guys write a reflection, take about 5 minutes or so, just like write down what you remember about how CRISPR works, how restriction enzymes work, and your own opinion on the ethics of genetic engineering. Thank you guys for your attention.